The topic for today is endophytism. Endophytes are organisms that can live inside of plants. So endo means inside, and phyte means plant. So inside plant makes sense, right? And I've been talking about ecology, and I've been talking about other sorts of subjects matter that have to do with endophytes and have to do with organisms and things like that that can live in and around plants. And when I talked about ecology, what's up, OG higher beings? When I talked about them in passing, I realized there was a lot of interest in that subject matter, and it's something that I find really interesting too, so I thought I would talk about it right now. So here I am. So most endophytes, so for one thing, endophyte research in general is, well, I guess we could say it's still in its infancy to a degree. Although we're definitely learning more and more every few months, every year we learn a lot more about microbials that live inside of plants and live around plants. So I'd like to first talk about like what it means to be a symbiont, or rather, what is mutualism, what is parasitism, what are all of these different like relationships that organisms have with each other. And that will help kind of illustrate the continuum of symbiosis. So on one side you have mutualism, and that would be characterized by a relationship in which both organisms benefit. But of course, we have to keep in mind that these designations, and every designation I'm going to go over, is from a human perspective and not always cut and dry. So mutualism is two organisms benefiting from each other. Then you have commensalism, in which one organism might benefit and the other one isn't really affected negatively or positively. And you've got mutualism, commensalism, and then you've got parasitism. And these are just kind of like general examples. There's different detailed parts of this continuum that I could go into more detail about, but I'm not going to. And parasitism is where one organism benefits and the other one is harmed. And you see in old literature, sometimes people will describe certain organisms as parasitic, and sometimes they don't. I guess that's sort of vague. What I mean is that, like, for example, I like to use the word pathogen instead of parasite, because parasite evokes a certain sort of, like, concept in people's minds that might not help them understand where I'm coming from. But essentially, none of these organisms know like that they are the good guys, that they're beneficial, from a human perspective. It's all from a human perspective and a cultivation perspective or an ecological perspective. So they don't know this, and it's really apparent because over a span of millions of years, you can have a parasite become a mutualist. In fact, you can have a parasite, like a parasitic microbe, become such a mutualist that they become a symbiont, living inside that organism, and transferred from progeny to, or from parents to progeny to progeny to progeny. Some microbes, some viruses even, become sort of like part of the genome, part of the genetic structure of an organism too. So that happens as well. So through association, a mutualistic organism over time can become less and less mutualist, and then become sort of there. And then that organism or that population of organisms can then become mutualist again, or more parasitic, or become a symbiont. All kinds of things can happen in this sort of setup. Anyways, my point being is that an endophyte if we're being super neutral, an endophyte is any organism that can go into a plant. So pathogens are endophytes too, but we usually just say pathogen because typically when we're talking about endophytes, we're not talking about things that can hurt the plant. We're usually talking about things that are either not noticeably affecting the plant in any way that we can usually tell outwardly, although they're still there, or we're talking about an organism that is benefiting the plant in a measurable way. And nowadays, within like the past decade or more, we understand that there are way more microbes out there that are endophytes to plants, and their effects are not always apparent to us, and their effects are sometimes quite beneficial. And I think there's a lot of focus and research in that aspect, particularly in the prophylactic capability of an endophyte for a plant. Plants have what's called an innate immune system. Humans and a lot of other organisms, animals, we have an adaptive immune system, and I've often described it as immune system 2.0 on the evolutionary scale, which is not, it's sort of an inaccurate metaphor, but it works for what I'm trying to articulate. Plants don't have neutrophils. They don't have white blood cells. They don't have specialized cells that are like moving up inside, up and down, and are produced by the plant that will seek out infection sites, seek out pathogens, and kill them through phagocytosis or through any number of other binding or signaling efforts. So plants don't have that. But what plants do have are endophytes and microbes, and sort of like an extended immunity, you could consider it. It's like immunity mercenaries that they sort of outsource, if that makes sense. So you have free-living microbes in the soil, for example, and they may come in, they might like sense that there's root exudates in the soil, for example, and they might travel along, make contact with the rhizosphere, make contact with the rhizoplane, which is the physical root surface, and then they will go and enter into the plant as an endophyte, or maybe just live on the surface of the root as an epiphyte. Epi meaning on the top of, phyte meaning plant. So that's possible as well. So, like mycorrhizae, for example, most people know what mycorrhizae are, and mycorrhizal relationships are usually mutualist. The fungus gets something out of the relationship, usually hexose or sugar, and the plant usually gets something from the fungus, usually like inorganic phosphate or other minerals that the plant would have to like mine down into the soil that they don't have to do as much for. And there's also water content. There's a lot more stability oftentimes. But I'll tell you what, oftentimes endophyte relationships or mycorrhizal relationships require the plant to be a little bit stressed, at least in nature. And those relationships are facilitated by plants that are stressed. If they're not stressed, they might not take to the mycorrhizal organism, or the mycorrhizal organism, you could say that, or that the mycorrhizae are, they would have difficulty kind of ingressing into the plant for a variety of reasons. And then finally, you had the fact that plants, the sort of plant microbe interaction oftentimes is like genotype specific. What I mean by that is that certain plants, certain species are going to be more receptive to other species, possibly because they, for lack of a better term, co-evolved with each other for millions of years. And they have similar sorts of genes and chemical exudates and signaling compounds and nodulation factors, for example. People who know that nitrogen-fixing organisms or plants they have to have the rhizobia, or they have to have the diazotrophs who um, take atmospheric nitrogen, and they can break that bond, that double nitrogen bond, which is very strong, and that's how they fix nitrogen. But for that to happen, the microbial organism has to there's this signaling interference or this sort of communication that happens between the microbe and the plant in the soil. And if that doesn't happen, if the nod factors aren't released and the plant and the microbe don't come to basically a chemical agreement, then that doesn't happen, and the nodules that the microbes will house themselves in and invaginate themselves into just doesn't happen. So it's really important that there's this sort of like genetic, I guess you could say, receptive capability, and it's not always there. And we're learning more and more every day that there are different things coming and going with regards to microbes and plants, and there are a lot of different factors that influence this relationship: environmental, but also genetic, chemical, and that sort of a thing. Pathogens are the same way. If you find a pathogen mutant, or you create a pathogen mutant that like doesn't have a certain gene that helps it infect the plant, then we find out that that gene was really important for, let's say, attenuating the plant's immune response. So maybe the pathogen really crucially relies on this gene to produce this enzyme or this compound that basically makes the plant unable to defend itself. But if it lacks that gene or several genes, because maybe it has what are essentially like what's the word I'm looking for? Like redundancies. So you could have a pathogen, especially a generalist pathogen, that maybe can affect a whole ton of different plants. So it doesn't just have one gene that can attenuate the immune system. It has like 4, 5, or 10, or 12. But if you get rid of them, or the most important ones, that pathogen is not virulent in the way that it was before. San Diego Cannabis Farmers Market, good morning. Cali Monster Garage, what's your opinion on adding microbes in indoor hydroponic, like cocoa or rock wool medium? My understanding is that a lot of times that's sort of, it's harder to establish. I wouldn't say it's impossible to establish, but my understanding is that a lot of times certain microbes have more or less difficulty establishing in that relationship. And again, in a lot of times, like in a hydroponic setup, the way that nutrients are being, the way that the nutrients are being irrigated, or that they're being provided, like I was saying before, a plant usually has to be stressed in nature to like have this sort of relationship. So if a plant is being well fed and well watered and not stressed in that way, it might not take up. But it totally depends on the microbe. It's really microbe dependent, and it's kind of hard to speak in general terms. I wouldn't say that it's like a bad thing, and I think the best advice with regards to that is to make sure that the person who's selling the microbes to you has, if that is in case the situation that you're in, that you're buying a product that's a microbial product or a supplement, just make sure they have data to back up what they're talking about. And if they don't, maybe like don't buy their product, possibly. I'm all about data-driven decisions with regards to that sort of a thing. Yeah. Mycorrhizae, for example. I'm going to be putting this video up on YouTube, like I always do. YouTube, Zenthanol. So if you want to know about more of that, I'll put in the description some examples of where in which we know that plants that are stressed take up the microbial relationship much better with endophytes, and those that don't, not as much. Another important factor actually to consider is that a lot of the elite cultivars that are like post-Green Revolution, the sort of crop cultivars we use now, a lot of them don't have the, like I said, they don't have that genotype specificity. They don't have the genes that essentially allowed for microbial interaction. And it's not like people intentionally outbred them or anything like that. But, if you take a population of plants from one location as humans and you go somewhere else where there's a totally different soil, a totally different sort of microbial composition generally, then those plants may or may not be receptive to those microbes. They may or may not have that reaction that they would have in another soil. So over time, those genes aren't important, and it's not like the people know that this is happening. It's not like the people understand that this sort of microbial relationship is happening back in early days of agriculture. So nowadays we know that happened, and we know that these genes are important. So there are efforts to kind of take wild cultivars of the plant and sort of breed them together, or in other ways develop the genetic architecture for this microbial plant relationship, which is pretty cool, and I very much support it. Dimebag Dozen says, have you kept bugs for pets and study? Absolutely I have. I have a couple of videos of a praying mantis, and which is Stagma mantis californica. Yeah, the California mantis. And I've also got a jumping spider. I used to have a Phidippus audax jumping spider that was really cute. And she died like a few months ago. But I could tell at that point because for several months she wasn't feeding, and she wasn't... she was very lethargic. So she was she lived a lot longer than she would have been, in the wild. I caught her when I was up in LA. I also used to keep Zophobus morio darkling beetles, or meal superworms. Actually, they weren't the morio, or yeah, they were the morio types. So the large superworms. Yeah, I've definitely kept them for study, for pets, kind of, just to kind of have something cool to do. I also enjoyed just feeding them scraps and just being good, so they would make waste, and then I would take that waste and I would add it to garden plant and things like that. And I like to do that kind of a thing sometimes. Do you know how they breed beneficial mites commercially? Yeah, it's different for different insectaries, but generally speaking, it is really, really labor intensive, very technical, for a couple of reasons. One, I've actually been asked this question a bunch recently, and I'm really happy because people are really interested in knowing how their commercial biocontrol agents are being created. Beneficial mites, for example, are very common, and so, for one thing, a lot of predatory mites look pretty much identical morphologically. If you're not looking through a microscope, you really can't tell the difference. That's why I get so many questions of people asking me, Matt, what's this bug? What's this mite? Is this a root aphid when it's really a hypoaspis soil mite or something like it? And it's really hard in an insectary to delineate that. If you don't have a really sophisticated structure that kind of houses all of these mites, and you're kind of keeping the contamination levels at a minimum, that takes a lot of effort. It takes a lot of money and takes a lot of time. Just curious why people don't do it at home. Right, so that's part of the reason why right there, is because you might not be able to keep your mites out of each other's cultivation spaces, so that's a problem. There's different strategies to producing different mites. So, for example, if you have a predatory mite that feeds on a living, obviously your living pest host, you don't necessarily have to feed it that pest. You can feed it an alternative food source. Or so like a different feeder mite maybe, or you could feed it like like not honeydew, but like sugar water for like parasitic wasps, or pollen for like Swirsky and omnivorous mites and things like that. So you can do those things, but some people believe that what you end up doing, and it's sort of true, you end up like conditioning the organism to not perform as adequately because it'll get used to the alternative food source. And this is where like microevolution comes into play. So over time, over a period of like many populations, you might not have an organism that's conditioned to like hunt in the same way, or to like search and seek out hosts like parasitic wasps do. If you breed the wasps with their food right next to them, then they don't get in the habit of like flying around and searching for their host. And if you do that over time, you will breed a less an inferior product essentially, because they won't be as adapted to, and they won't be as sensitive to, in the very minute ways, to search for their host. So that makes sense. So basically, you really need to know what you're doing. Yeah, you really do. I've had several people come to me and ask, like, "Hey, like, let's, why, can I just make predator mites myself? Like, why do I have to pay this company?" And you have to pay the company because, one, there's a bunch of laws regarding, like, the trafficking of insects and predatory mites and all that kind of stuff. So that's a big deal. For example, like Koppert Biologicals in the Netherlands has to produce 2 different bumblebee species for their Natropol system, I think it's called. But like they can't use one of their bumblebee species for America because it's exotic, and the organizations that are in charge of such a thing don't feel comfortable introducing yet another exotic organism if they don't have to. Sometimes it's inevitable. It's not as bad if it's like a specialist, but again, there are permits and things that are part of this sort of arrangement, which is good ecologically. Like I've said, it's important to like consider the ecological ramifications. Otherwise, you'll have the cane toad problem, and if you're not familiar with it, you should look into it because it's a great model example of why you don't just willy-nilly add organisms to the local ecology. Fishy420 Dab says, "I think it would be awesome for you to do a segment on using predators to annihilate pests indoors by using a ton of beneficial predators. Overwhelming the predators as opposed to outdoor where this would not be possible. I feel like if you put a crazy high number of predators, you can possibly rid yourself of pests like thrips." So that's called inundative releases. There's 2 kinds of general ways of using biocontrols. There's augmentative releases, and there's inundative releases. In augmentative releases, you want to establish the organism, so you only put out an amount that you need to establish a population. And when you do that, you are expecting the predators that you put in to establish and to have their population grow. And that's one way. So that's augmentative. And then you have the inundative, where in which you just like blast an area with a ton of predators, and you're just using sheer numbers. There's a really cool video that I made on exactly this subject matter, and it's in my IPM Intro to IPM series. And go onto YouTube, Zenthanol, and check it out, because I go over exactly that topic, because it's really important to consider your strategy when you're using beneficial organisms. It's more expensive to just use more organisms, but it might get the job done quicker. And depending on what your resources are as a cultivator, it might make way more sense to do that instead of waiting for them to establish. That's more of a preventative measure than a curative one when time is of the essence, which is most of the time. Hashmelt says, "My bug rep told me we couldn't use pirate bugs for thrips because our sprays would kill them. We sprayed Dr. Bronner's and Green Cure after week 2 of flower. Now, Does this sound right? We use Swirsky now. Yeah, I mean, I could see how like spraying something like Dr. Bronner's might incapacitate predator bugs like pirate bugs, like minute pirate bugs. Um, I mean, how do I put this? It might not be cost effective. That could be like the more nuanced thing. I don't know what your cultivation setup is, and that's another important thing, is that making generalized statements without knowing the context of the cultivation space uh, can be fallacious. Because if I don't know that you're larger or smaller, or if I don't know um, like what your resources are, like for example with the inundative releases, if you if you have a spider mite population and maybe like 30% of the crop is affected, but it's very small and spread out, you could use X amount of Phytocilius persimilis predator mites to affect that spider mite population. Or you could use X times 3 mites and maybe be quicker, but it'll cost 3 times as much money, or like 2.5 times as much money, depending on the relationship you have with your insectary or your beneficial insect company. So that's probably what is going on there. But I can't really comment because I really don't know all of the context. But that's a possibility. So yeah. So that's sort of what I'm talking about here. That if you're there's different strategies. Check out the IPM video. There's also a few videos I've made on different biological control agents, and I've discussed this often, where like when I'm assessing a biocontrol agent for its efficacy, like against broad mites for like Swirsky and Californicus and things like that, or for russet mites and things like that, and I've got videos on this subject, it's really important to consider things like what's called the attack rate, or which is sort of an ecological term, or the like intrinsic rate of natural increase, which is a complex algorithm that essentially tells you at X temperature with with certain variables what is the intrinsic rate of natural increase, which is how many females per female does the population produce, because the females they control how much more of the organism you will have. So if like under certain conditions the rate of increase is like 2, then that means that there are 2 females being made for every 1 female. So you have a replacement and an additional female. Or you could have something massive like 15 females per female, depending on a different food source or a bunch of other factors. Dimebag Dozen is asking me what my favorite bug type Pokemon is. And that's not hard for me to answer. It's definitely Beedrill. I like the Weedle evolutionary line. I have many fond memories of Generation 1 Pokemon going up and using Weedle and Beedrill. Not a very powerful Pokemon by any stretch of the imagination, but I really enjoyed that evolutionary line. I thought it was really cool. I even liked the sound effects that they had. It's funny you mentioned that, because I have for my phone, for my my ringtone for my phone is the Generation 1 Starmie cry. I also have it as an alarm because it's really, really obnoxious, sort of. It's kind of an odd sound. You guys can look it up yourself if you want on YouTube or something, but yeah, it's funny you mentioned that. Let's see. But I was going to talk about endophytes. So endophytism is a really cool thing, and I'm really looking forward to some of the research regarding using endophytes as sort of like I was saying before, a secondary immune system, because although plants don't have an adaptive immune system like us, where you've got like little microbes that we produce, like cells, white blood cells that are traveling up our circulatory system and killing bad stuff. Plants do have other organisms that kind of come in and happen to do that. I mean, again, they're not like communicating like there's an intelligence there, and a lot of people don't agree with that, and I know that's sort of a controversial thing for some people, but that's not really a belief that I hold. But it's not necessary for there to be like an intelligent conversation going on. What's necessary is for there to be a relationship going on at all. And I don't think that it detracts or is I don't think that's a negative thing to say that, because it's still happening and it's still beautiful in that way. It's almost like it's more beautiful because it's automatic and there's no like higher sophisticated functioning going on. It's like more primal in that way, I suppose you could say. Yeah, so Endophyte Research, mycorrhizae, other organisms that can like go into the seed, and then they're in the seed packet, and then you plant the seed and they're already there. Pathogens can do this too. There are definitely seed-borne pathogens like viruses, and botrytis, for example, can be seed-borne for some species. I have a post about that I made recently. So it's really cool. It's really complex. It's really hard to make a ton of generalist statements because I'm sure some of them will not be true in some cases, and you're going to be the one stuck holding the bag if you say one thing and then later on the research comes out that that's not true. So I want to make it really clear that I'm going to try to talk in generalizations as much as possible without being like lazy and indolent. Fishy420 Dab says, a little clearer follow-up question. Do you think it is possible to completely or very closely eradicate a pest indoors using predators? Yeah, absolutely. Especially because it's indoors. Especially. It depends on the predator, it depends on the pest, but generally speaking, yeah, I would say that that's usually what you're trying to do. Because if you have a closed-off space, and as long as you kind of the pest got in some way. If it got in because there's like a structural deficiency, like a hole or something about the ventilation or something like that, then you can have that problem. But if it came in like phoretically, in other words, if it came in on a person's clothing, some equipment, some new plants that came in from like another country or from like your buddy or something like that, then that can happen. But if you have like a closed off space like a room or something, then that's your battle space, right? And you figure that as long as this goes back to what I was saying about attack rate, so and intrinsic rate of increase. If you've got a predator, and this is why life tables are important. I have a video on my YouTube channel about life tables and how to read life tables a little bit, kind of a general primer, because some beneficials do better in different temperatures and humidities, and pests do better at certain temperatures and humidities. So what you end up having is a situation where the predators that are good biocontrol agents have a high attack rate, which means that they attack or kill a lot of organisms during their lifetime. And if you have a room where you have X amount of predators or pests and they reproduce at a certain rate, and you have predator that comes in and it reproduces at another rate and it goes higher than that pest, then that's good, because once you reach this sort of breaking point and it starts to increase, then the pest population will start to decrease quite a bit, and you have this sort of like W graph going on. So in a room where you're not getting reinforcements from other pests coming in, and you only get reinforcements from predators, then if you inundatively release, like was being asked before, you can just increase that sheer level of population and that intrinsic rate of increase. But if you only use a certain amount, which is cost effective, then that's okay too. It's just that like there's a what's the phrase? There's not a return on investment after a certain point. You can really explode your indoor garden with all kinds of predators and or a huge high number of that predator, and you can decrease the population of pests really swiftly, but it will cost you maybe 3 times as much. So you run into that problem logistically. How do you put the hurt on fungus gnat larvae? It depends a lot on what you're growing, and depends a lot on how many there are. But I like to use Bouveria bassiana as a drench. I think that's a really cool way to use a biological control organism that will go after the fungus gnats. I'm not a huge fan of nematodes, but that's just a personal preference. I know many people who have used them successfully, so I have nothing bad to say about nematodes. Um, Pyrethrin compounds can be good. And I'm speaking generally for anyone who's like, oh, I can't use that in my state, or I can't use this, or whatever. Like, whether you're a gardener, or a farmer, or growing cannabis, or whatever, there are all kinds of ways to get rid of organisms. But I would say it's really important to focus on the larvae and the eggs, and not so much the adults. And also, culturally, in your environment, it's really important to not allow the population to grow out of control, but it's also important to not keep your soil too moist, because that's a big problem that people do. They overwater, and they might even have a little bit of root rot or something like that. I want to make sure I get this question somebody asked. Hash Melts is like, we got rid of russets by putting 8 sachets per plant. Right. Yeah, that's sometimes you just got to do that. You just got to put a whole bunch of the biocontrol agent out, and that's really your only choice in the matter. But in other cases, you can rely on a preventative amount first, and then supplement it with a curative release later if it becomes problematic. Great, I thought so. If you put enough predators, awesome. Infoman, you gonna be rich one day. Well, I appreciate that. I hope you're clairvoyant. Andrew Giebelhaus says, could you talk about biological control for spider mites? Yeah, I can. I have a few videos on my YouTube channel about this subject, so if you want to learn more about that, hit up my Zenthanol YouTube channel, which link in the bio or whatever, as the people say on Instagram. Spider mite control has a lot of options because spider mites are very, 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 very common. So you've got the spider mite fly, Acarasuga felchiae, or did I get the genus and the species mixed up? The spider mite fly, which I have a video on, is one example, though I don't think it's very popular. There's also the Phytocilius persimilis, the type 1A spider mite, or predator mite, which is the king of spider mite control, in my opinion, as if you're using a biocontrol agent anyways. It's co-evolved with the spider mite over millions of years to be effective at cutting the silk, traveling on the silk, feeding on the eggs, feeding on the adults. So it's literally made to work really well. Those are some examples of biocontrol agents that I think work really well, and you're very welcome for that. Integral tilth? Integral tilth, I like that. Any way to deal with root knot nematodes without discarding soil? Probably. I'm not very good at getting rid of root knot nematodes, to be honest. Most people aren't. They're difficult. They're difficult pests for a reason. There are fumigation possibilities for control, but even that doesn't work very well. I guess you could try to use a entomopathogenic fungus or something, but honestly, there aren't a whole lot of good options, which is why chemical agents are so commonly used in agriculture for the stem rot and the root knot nematodes. Because they are just that difficult to deal with. There aren't a whole lot of good options, so I don't really have an answer. That's one of those examples where sometimes I won't have an answer for you, unfortunately. Sorry. Um, yeah. So, anyways, that's those are endophytes that I was talking about before. But obviously, there's a lot of interesting conversation regarding biocontrols. I'm glad people are interested. You wouldn't believe the amount of people that I've worked with in traditional agriculture who are very, very skeptical of biocontrols, which is understandable because for a long time it was sort of difficult to guarantee quality control, and now it's a lot easier to do that. Our understanding of the relationships with biocontrols and pests is a lot more sophisticated, so it's a lot more better. But unfortunately, there's still that stigma, and also there's a cost factor. You're not going to have a lot of soybean growers with millions and millions of acres shelling out a lot of money for biocontrol agents as long as they are priced the way that they are. So that's just kind of how it is. I think I'm going to leave it at that though. If you just hopped in or you just missed the video, then you can get this video on my YouTube channel. It'll be coming out, probably scheduling it for a week from now or something like that. I will answer one more question though, since I'm getting it. General Mombi asks, the soil drench to combat fungus net larvae was blueberry what? Beauveria bassiana. I have videos on that. I have lots of posts on Beauveria bassiana. Some of the products that are easier to pronounce that have this fungus in them is Botanigard or Mycotrol, for example. So take a look at that. Check out my YouTube for more information on Beauveria bassiana, biocontrol agent usage, integrated pest management, all of that sort of information. Because it's really important for people to have access to that information, which is usually behind a paywall. So check it out.